Hello, I'm Michael Lawless, Senior Principal Scientist at Simulations Plus. Welcome to my presentation on AI-driven lead discovery, where I'll showcase our AIDD module in ADMET Predictor. I want to acknowledge my co-authors, David Miller and Marv Waldman, who programmed the AIDD module in ADMET Predictor. This slide is an outline of the presentation. First, I'll discuss computer-aided de novo drug design background. Then I'll give an overview of the AIDD module. I'll discuss how we create our candidate molecules through SMERX transformations. We have a synthetic feasibility score. We can incorporate pharmacokinetic and ADMET liability predictions. We penalize any models that are out of scope. We use capping values, and we can also call external programs to compute objective functions. In the second to last section, I'll discuss how we select the Pareto optimal compounds to go into the next round of AIDD. And then finally, I'll demonstrate our AIDD module technology. This slide discusses the history of computer-aided de novo drug design. It started out in the early 1990s when people were interested in structure-based design, so using a crystal structure of the receptor or enzyme. And there were programs such as LUDI, MCSS Hook, and Sprout, which essentially started with a fragment in the active site and then would grow that fragment by adding methyl groups or fragments to the molecule and scoring them. In the late 1990s, Lipinski introduced his rule of five, and so people started talking about ADMET properties and that drug design was a multi-objectional process. So it ain't just about activity anymore. So the days of just optimizing the activity were over. You needed to actually get the compound into systemic circulation. Then in the early to late 2000s, we started to see multi-objective ligand-based and structure-based design. And here, typically, the multi-objectives were combined into a single function. You had programs such as EA Inventor, Muse, and then pharma companies started creating their own in-house programs. In the last decade, Pareto-based optimization has kind of replaced this single function, and deep learning generative algorithms have started to come into fashion. About 3 or 4 years ago, there were very few publications, and within the last few years, there have been many generative algorithms that either try and modify the SMILE strings or come up with new SMILE strings or molecular graphs. This slide addresses generating and selecting or scoring virtual molecules. So we start over here on the left on generating virtual molecules. There are ways to just do simple transformations, elementary transformations, so modify the molecule with small changes, turn an oxygen into a nitrogen, etc., or add a fragment or delete a fragment. You can also add common fragments, for example, replace a carboxylic acid with a tetrazole or other bioisosteres. Here you have to concern yourself with synthetic feasibility. And early on, this was based on filtering the molecules on structural alerts. For example, if you generated a hemiacetal, you would have a smart string that would flag those type compounds and remove them from the pool of candidate compounds. During the late 90s, when combinatorial chemistry started becoming a fashion, people were designing combinatorial chemistry libraries based on reactions and using known reagents or reagents that you could access quickly. With these libraries, you had to worry about diversity. Certainly, if you had a scaffold and you were enumerating the scaffold, many of the compounds would be similar to each other, so you might need to do some kind of diversity selection there. And these could be SMERC-based, so these SMERC-based transformations would encode the synthetic libraries. And then more recently, deep learning algorithms that generate molecules based on the SMILES or graph representations there. Now, once you've generated molecules, you need to score them. We mentioned multi-objective criteria, combining some of these into a weighted sum or combining function, selecting the Pareto optimal subset. And so this is what we use in our procedure. Then you also have to look at the criteria for selecting these molecules. You want to based on the activity or docking scores, ADMET liabilities, sometimes similarity to a known lead, but you don't want to get too close to that need. So you need a little bit of novelty in there. You certainly have to address the synthetic accessibility of these compounds. Make sure they have they're drug-like. There's no peroxides or epoxides within the molecules, and then using chemical filters to extract those. Now I want to specifically start talking about the AIDD module in ADMET Predictor. The goal here is to design compounds that have high potency and selectivity at the primary target, high synthetic feasibility, and good ADMET and pharmacokinetic properties. This can be used in the hit finding, lead optimization, and scaffold hopping phases of the lead discovery process. This slide is an overview of the AIDD workflow. You have to start with some kind of knowledge base, and this could include crystal structure, either an apo crystal structure or inhibitors or bound or ligands bound into the active side of the crystal structure. Any SAR information you can glean, and QSAR model could be used as input. And this is going to develop as you develop in the process. Next, you select a seed molecule or a set of molecules that you want to optimize. Then we generate candidates, and this is done through the applying Smirks transformations to randomly selected compounds in the current population. Once it converts a molecule by applying a Smirk string, it looks at what we call a scaffold query and makes sure that it abides by that scaffold query, meets the requirements of that. And these would be the requirements, the structure activity requirements that you've gleaned so far. There's also a drug-like filter, and if the candidate compound hits or matches one of the smart strings in there, then the compound will be rejected and we'll have to generate a new compound for that. So drug-like filters will be acetals, Michael acceptors. You can also specify simple properties such as less than or equal to 4 aromatic rings in there. And of course, this is modifiable within our program. For example, I talked to someone last week that indicated that they were working with Michael acceptors, so they wanted to form a covalent bond with the receptor, and so they wanted to keep Michael acceptors, obviously. Next, you evaluate the properties of the molecule. So you apply your QSAR model. You can also dock the molecules or use ligand overlays. Score it with synthetic feasibility. We typically recommend that. And then ADMET Risk. And then we can use our HTPK module to calculate things like oral bioavailability. Next, all the compounds go into the Pareto optimal scheme here, and it selects the best compounds for the next iteration. So you're always keeping the pool, the compounds in the pool that have the best overall scores for these various molecules here. And then you simply iterate over and over again. This slide illustrates how we generate molecules using Smirks transformation rules. So you take a simple molecule here, and we have a file that contains these transforms. So these can be simple isosteric replacements, reactions from literature, in-house experiment, and then what we call these chemical intelligent mutations. And this comes from a paper from Stewart et al. Of Abbott in 2006, and they created a scheme called Drug Guru, which would make kind of medicinal chemistry modifications to a hit or lead structure. Users are allowed to modify the default rules. So basically, you're just taking a structure and transforming it into other compounds here. So for example, this top one, it moved the nitrogen from this position over into the bridgehead on the bicyclic ring. Now we want to perform these transformations in a chemically intelligent manner. In other words, we want to try not to generate non-drug-like structures. That just simply slows the process down. If you generate these, you can always catch them with the drug-like filter, but it's best not to generate those type molecules in the first place. So here we have example of converting a non-fluorine to a fluorine atom. So a simple Smirk string would take anything that's not a fluorine atom. So fluorine has atomic weight 9, so this is specifying a fluorine. So then this bang says it's not a fluorine. This non-fluorine atom has to be kind of on the end of a molecule and then convert it to fluorine. The problem is, and what you need to avoid, are structures like this, where you take a carboxylic acid and apply the Smirk string to this OH or group and replace it with a fluorine. Now this is going to make a reactive, I would call this an acid fluoride. Certainly if it was a chlorine, it'd be an acid chloride. But that's reactive and you wouldn't see those in drug molecules. So you'd want to modify the SMERC string further so that it avoids carbons that are attached to a carbonyl here and then replace those. Currently in the AIDD module, we have 150 transformations, and it generates a vast array of compounds. Now let's turn to the type of properties we want to calculate. So this is the first dialog box within our AIDD interface, and we've selected synthetic difficulty here, oral bioavailability, ADMET Risk, BACE1 model. So this is a QSAR model to predict the PIC50 of BACE1 inhibitors, and a BBB model. We have over 50 built-in models, including oral bioavailability, fraction absorbs, synthetic difficulty, and then user models. You can also click this button and apply any objective function you can compute with a third-party software or an external application. And then I'm also going to talk about the direction that you want the molecule to improve onto. So with our synthetic difficulty score, higher values mean it's harder to synthesize, so we want to minimize that. So you're directing the Pareto optimal scheme. In the lower right-hand corner of the dialog box, you can specify the pH for pH-dependent properties such as log D or aqueous water solubility. You can also specify a dose for the pharmacokinetic property prediction. I'll discuss the details of these property predictions on the next few slides. This slide discusses our synthetic feasibility assessment. It's based on a journal article from 2009 from Ertl and Schaffenhauer. It basically scores the molecule based on two components, a fragment score and a complexity score. The fragment score is just a measure of the fragment frequencies. So we downloaded a large number of compounds, 1 million compounds from PubChem, and we calculated ECFP fingerprints or keys for each one of the compounds. And then we measured the frequency of those fragments in the data set. Then when you take an arbitrary compound, you break it up into these fragments, and you see how many of the fragments have a high occurrence within PubChem. And the idea is that does the proposed compound contain fragments of compounds that have been synthesized? If so, it's more synthetically accessible. Then we also penalize for larger number of heavy atoms if there are macrocycles, stereocenters, spirocenters, or bridgehead atoms. This is a comparison of our synthetic feasibility score versus the assay score from Ertl's journal article. And it's based on some of the molecules that were published in the article. Here are a few of the differences. We used a larger number of compounds to train the models, and then some subtleties within how things were defined as far as the ECFP fingerprint keys. And our range goes from 0 to 10, and the higher the number, the more synthetically difficult it would be to make that particular compound. This next slide describes our high throughput PK simulation, and this is how we calculate the oral bioavailability of a compound. And one of the things that makes AIDD module unique is the capability to use PK predictions within the as an objective function within the program. It starts over here on the left, and we simulate an immediate release tablet either in a rat or human species. You could also do an IV bolus. This slide, of course, addresses the oral dose. So the compound would go into the stomach and it would start dissolving. Here are things like the pKa of the compound, the solubility of the compound, the aqueous water solubility, passive and fessive solubility would determine how fast this compound dissolves in the stomach. Then it would start working its way down the GI tract where the parts of the compound that are in solution could be absorbed into the gut wall. And once it gets into the enterocytes of the gut wall, this is a term, the fraction absorbed of the compound. Next, it would go into the liver where it's metabolized, and then whatever comes out on the other side of the liver is the oral bioavailability of the compound. And we use our models to predict solubility log P, which is important for the uptake of the compound, human jejunal permeability, and other parameters in this model. This slide discusses our ADMET risk parameter. So it's a composite scoring function that the higher the value indicates the more risk to develop the particular compound. ADMET risk is a similar procedure to Lipinski rule of 5, in that we took about 2,200 orally bioavailable compounds from the World Drug Index, and we measured a property such as molecular weight or the number of heavy atoms, etc. And then we set cutoffs where 90% of the compounds would pass that cutoff. So, for example, molecular weight, the cutoff starts at 450 and goes up to 500. And this is kind of a linear scale. So the penalty would start at 450 and then linearly increase to 500. And then at 500, it would, anything above 500 would get a penalty of 1. So we have various parameters similar to Lipinski scores to look at the absorption, but we also include permeability, aqueous water solubility. So we have more rules than Lipinski did. We also include a rule for the volume of distribution, toxicity scores such as hard liability or acute rat toxicity, hepatic toxicity, mutagenic risk, and then if the compound is quickly metabolized. This slide shows how we define our applicability domain for all of the models we calculate. And it's just simply based on the minimum and maximum value of the descriptors in the model. And in this example, we have a model that's based on only 2 descriptors. So we have descriptor 1 and 2. And we know for the training pool all of the values of these descriptors, so we can calculate the minimum and maximum value. And then additionally, we apply kind of a 10% border around this minimum and maximum value. And then any compounds that are predicted there whose properties are within this range would be in the applicability domain or in scope of the model. And then any molecules where their properties fall outside of this would be called outside the applicability domain of the model. And in the interface, these are highlighted in magenta and underlined. Now, as far as AIDD is concerned, if we predict a property that you're optimizing, such as BACE1 PIC50, and that prediction is out of scope, then we would apply a penalty to that prediction. So we'd lower the priority of that molecule, and so it wouldn't be typically on the Pareto front. So this way, we kind of eliminate molecules that have out of scope predictions. On the flip side of the coin is something we call capping values. If we look at these compounds, they're very easy to make, so their synthetic difficulty score is very low. So these would be on the Pareto front and would be selected by the algorithm based on synthetic difficulty. And you really don't want these trivial molecules that they're very good to make, and they're good in one objective, but they're just not going to have very high property values as far as activity. So we came up with something called the capping value. And in this case, we applied a capping value to the synthetic difficulty. And what this argument here means is that if the synthetic difficulty is below 2.5, then we reset it to 2.5. And so all these compounds would be scored as the objective score would be 2.5. And other molecules that are have that similar synthetic feasibility would also be on the Pareto front. So you would kind of eliminate these trivial compounds. So we also apply it to things like the oral bioavailability of a compound. If it's above 90, that's really good enough. It's not, you really don't want to weight one that's 95% compared to 92%. So these would all be put on the same level. And as far as synthetic difficulty, it allows us to filter out these trivial molecules, and it doesn't encourage the algorithms to go into simplistic molecules. So far, I've focused on models and properties that can be predicted within ADMET Predictor, but there are obviously some things that we can't predict predict. For example, we can't dock a molecule and score it in an active site. So for this, we suggest using an external application, and this is done through the first dialog box. You would simply click this, compute objective functions using an external application, and then you would have to specify a text file that we provide a template for the external application. This next slide outlines the parameters in the external file. So first, there's a file path alias that you would set to a folder or directory, and it also defines this variable called path that's enclosed in square bracket. And when I run AIDD with an external application, which I'll demo at the end of this talk, I typically set this variable to the AIDD output directory. That way all the files from the external application and the AIDD run are in one directory. Next, you have to specify a script or an executable. The variable name is a command line, and it points to a script or an executable program. We can do a bat script or a shell script or an executable program. AIDD, once it starts running, will output a structure file, and this is specified in the external file. The variable name is structure file, and it points to file name. And typically I include the path and then the name of SD or SMILES file, which is created. So the options are SDF or SMILES file. And then you can also optionally output ADMET predictor parameters such as log P or other descriptors that may be used in your external program. So those would be output in this file here. Next, you specify the name of an output file, and this is the file that's created by the external program, and it contains the scores of the objective function. So for example, if it was AutoDock, it would be the docking scores for each of the compounds, and they would be, if you can have more than one objective function generated by your external program. If so, there would be one line per molecule, and then it would, you'd tab separate the individual fields in there. The next parameter is an objectives list. So this output file is a text file, but there's no header information. So this is essentially the header, and it's the name of the objective function from the external program. For example, AutoDock underscore score would be one of the potentially an objective list. And then for each one of the objectives, you have to specify if this property is minimized or maximized as the preferable value here. Again, this would be comma separated in the parameter file if you had more than one objective function, and similarly this would be comma separated, and it would be in the same order as this file. Now, once AIDD will, when it hits the external program, will wait until this script finishes executing. And then it will check to see if the number of lines in this file is equal to the number of output file molecules that were output in the SD or SMILES file. If not, it's going to issue an error message, and these files would stay around. If everything is read in correctly, then these files get eliminated and then are used in the next iteration of the program. Now, I've mentioned Pareto optimal front several times in the talk, but I haven't gone into detail to explain what a Pareto optimal selection is. And it's illustrated by this plot. So here we have two objective functions, x and y, and lower values of both of them are preferred. So a Pareto optimal front is defined as a compound where no other compound is better than it in all directions. So for example, this first point over here in red is better than any other point in the x direction. It's got the lowest x value of any of the points in this display. And here these would be molecules that are scored there. So that would be on the Pareto front. Now if we move down, this molecule or point is not as good in the x direction, but it's better in the y direction. So these two would be on the Pareto front. And then similarly, you can work your way down through all of these red dots and justify why they're in the Pareto front. For example, it's better in one of the directions than either of these two molecules. So these would be all on the Pareto front, and they would go into the next iteration for AIDD. The rest would be eliminated. Now, there is a point within the AIDD optimization where you can specify to keep more than a certain number of points. And so if the minimum number of compounds in the pool is below what you want it to, then it will select additional compounds on the Pareto front. And so it will eliminate, essentially, not work with the ones in red, and then select the next ones, which are on the green ones here on this Pareto front. And then those would go into the pool. They'd be mutated. Those new mutated molecules would be scored then. In this next section of the presentation, I'll demonstrate setting up an AIDD run. I'll use a retrospective example of discovering BACE1 inhibitors. BACE1 is an enzyme in the brain that cleaves an amyloid precursor protein that eventually forms amyloid beta proteins and is hypothesized to be a crucial step in Alzheimer's disease. There have been compounds nominated for clinical trials that are BACE1 inhibitors, and they've failed either for futility or safety reasons, as this quote mentions. But this quote also says that it's still a well-validated target for Alzheimer's disease, and that there's still potential to find a candidate compound for this. So this is a publication, a recent publication in 2001, almost a year ago. And this is just the crystal structure of BACE1. And what I'm going to do is use an external program to dock candidates from AIDD into this active site. I would just want to acknowledge a few of the programs that I'll use. So I'm going to use AutoDock to perform the docking and scoring of the molecules. I use Yacera to visualize the docking poses and also set up the protein for docking, so defining the active site in there and cleaning up the protein. I use Babel to convert from one file format to another. And all these programs are freely available on the web, and we'll use them as an external application along with internal ADMET Predict models. We're going to dock into a PDB file 6FGY, which is the BACE1 inhibitor with a ligand bound into it. And we're going to use molecules from this Rambot article from 2015. And we're going to try and reproduce some of the basic molecule from this publication. This next slide simply shows a workflow for the AIDD AutoDock example. So the first phase of the external program will be, it will generate candidate molecules from AIDD. These will be converted to a dominant microstate. This uses ADMET Predictor to predict the dominant microstate at a specific pH. So this uses ADMET Predictor command line. We also have a program that we're going to release in the next version of ADMET Predictor, which will be due out this spring, that converts molecules from 2D into a 3D structure. Then these molecules will be used as input to a program called prepare_ligand, which is supplied by the AutoDock program that defines rotatable bonds and charges on the ligands. Then I'll use ADFR, which is part of the AutoDock suite, to dock the molecules and score them in the active site. Then these results are returned to AIDD, and the Pareto optimization is computed. I've started ADMET Predictor and read in a seed molecule here. So this is an oxazine-containing ring molecule. It's similar to molecules from the Rambot's article. We have to specify the chirality around this setter. That's important for binding into the receptor. And what I want to do with this AIDD run is kind of explore the shape of the ligand in order to fit the pocket the best. I'll show on the next phase where my scaffold query only allows substituents in the ortho, meta, and pair position of this phenyl ring. So to start the AIDD run, you would select a molecule or several molecules if you want to include more than one seed molecule, and then you click the AIDD icon button up here. So we're going to use synthetic feasibility, oral bioavailability, ADMET Risk, BB Risk, and we're also going to use an external program to perform the docking. So this is going to be use docking as one of the scoring functions in the as an objective function. So here we want to select this external file here, and I'll discuss that in a second. Let me just go down the list of objective functions here. So synthetic feasibility, we're going to set a capping value of 2.5. So anything below that would get adjusted back up to 2.5, and lower values are better. For oral bioavailability, we're going to set a capping value of 90%, and then I'm also going to increase the dose up to 100%. Now these molecules are pretty soluble because this ring here can potentially be protonated, or is protonated in most of these, and so the solubility is reasonably high. If there was solubility issues, maybe we'd see a difference in the oral bioavailability between 0 or 10 and 100 milligram dose, and that would mostly be due to differences in the fraction absorbed. We'll use ADMET Risk. This contains all our risk values for absorption, distribution, metabolism, and toxicity. BB Risk is a special model that we created for this particular demo. It includes our BBB filter and log BB models that are in the PhysChem and biopharmaceutical module. It also includes a penalty if the compound is a PGP substrate. So if it's a PGP substrate, it probably won't get into the brain because it gets pumped out. And that's in our model in our transporters module. And then also we included a penalty for excessive positive charge. This comes from the Rambod article where they indicated that compounds with higher charges didn't have the correct distribution within the body or animal. And so they wanted to lower that. And they also think that if it's a higher charge, it's more potential for being a PGP substrate. Then as far as this external application, so what we're going to do is we're going to use AutoDock to dock these molecules into the 6-FGY crystal structure and score them. And so that's going to be another objective function in addition to these 4 I've already specified. So if we take a look at this particular file in Notepad, here's your file path alias that I talked about on one of the slides, and I'm going to set it to a unique directory name. I'm going to call this AutoDock 100. And then we also, for the command line, we're specifying a BAT file. And it's a very simple BAT file here, so I need to save that. And then if we take a look at the BAT file, it simply changes directory into this working directory. So we're going to, again, change this to 100. And then it's going to execute this script, AutoDock v8.python. Which is shown here. I'm not going to get into the details about this, but there's a lot of comments up here. First thing it does is it can define a capping value. So we're going to use a capping value of minus 8.1 on the docking scores. And it will read the SD file from AIDD that was specified in this text file here. And then it converts it to the proper charge state. So it uses our pKa models to protonate this compound. And in most of these, this nitrogen is going to be have a pKa above 7, so it'll be protonated in the majority of the compounds here. So then I just simply click on the next button. Now here's where we define our scaffold query. So I'm going to load this from a file. And this is a scaffold file, scaffold query. It's a smart string that specifies that you need an NH2 group that's bound to a ring carbon or carbon, then a CH2. So specifying that this has to have 2 hydrogens on it here is going to fix these 2 positions along with the oxygen, the nitrogen. And then when we get to this carbon, it has the right chirality here. And then as far as this substituent here, the first 2 atoms have to be degree 2, which means it can only have 2 bonds to it. So this is essentially blocking substituents on this and only allowing for ortho, meta, and para substituents. I'm also going to specify a specific drug-like filter that I set up. And so it's called BACE1 drug-like filter. And I can show just a couple lines here in this file here. So this query here specifies 3 substructures essentially, and it's meant to only have one substituent on that phenyl ring. So here is the ortho and meta positions on the ring. So if you have substituents there, it's going to reject it. Similarly, if it's ortho or para and then meta and para, it's going to reject those. So we're only going to allow one substituent on this ring in this initial exploratory phase of AIDD. I'll click the next button, which goes to the final dialog box in AIDD. Here I'm going to browse for specific transform rules. Now, we have this default molecular transforms. We modified these slightly for this particular run, and we're going to include those modifications in the next version of ADMET Predictor. So I would come here to the support files and then specify these molecular transforms here. We can also block certain transforms, and I've saved those in a file. So I don't want iodines in any of my molecules. I don't want nitro, so I've eliminated some of these. So I'll load this from a file called BACE1, again in support files, disabled transforms. And what it's done is it's marked some of the transforms as not appropriate or won't be used from the defaults in that file. Then I'll click OK. We're going to start with, we're just going to do 50 iterations. We're going to do 25 candidates per generation, except for the first generation. We'll generate 50 molecules. And then the minimum size of the population is going to be 25. Next, we have to specify the output folder. So I'll browse to the subdirectory here. And then I'm just going to create a new folder. And I'm going to call it AutoDock -100, and then CD or change into that directory and select that folder. And then I would click run to get that run going. Now, if we were just using internal ADMET Predictor models, this would fly by. This would only take a few minutes, but AutoDock takes about 15 to dock a molecule, 15 to 20 seconds to dock a molecule. And so that is the bottleneck for this actual run. And this would take, I think, 6 or 7 hours to run. Okay, so instead of running that, I'm simply going to cancel that out. Now, let's take a look at the output directory. Now, this is from a previous run, so AutoDock 09. But the script, what the script does is it creates a folder for each one of the iterations from AIDD. So there's going to be 50 folders here named AutoDock. And then in each folder, this is going to hold the results of that particular AutoDock run. So it contains all the input and output files used for AutoDock. And then I also have a script that goes out because the Python script saved the AutoDock scores in a file that was imported back into AIDD. It also creates a file that contains the molecule name and the docking score. And so I ran a script to kind of combine all those into a single file called all AutoDock scores. And then I'll import this into the results from the AIDD run. I also have a script that runs and it creates a BAT file so that we can visualize the docking output or the docking poses for each one of the compounds. So this is the output file from AIDD, and I've imported the raw AutoDock score. This is the objective function that was specified in AIDD, and note that there's a capping value of minus 8.1. So anything that scored above that was set to 8.1, and that was used in the Pareto optimization. I also have created a cell that contains a BAT file within ADMET Predictor. If you have a link to a file, you can click on that and it'll run that program. So it's executing this BAT script that starts yes, Sarah, so that we can visualize the pose here. And in green is the co-crystallized ligand, and then colored by atom type is the docking result from the AIDD structure here. And you can see it overlays pretty well on top of the co-crystallized ligand, and it matches these hydrogen bond donor acceptor pairs in here. So let's turn that off for a second and then show the H bonds within the active site. So here you're seeing the two aspartic acid residues here that are making salt bridges and hydrogen bonds to the oxazine ring. Then you also get a hydrogen bond from this nitrogen, this amide group, to carbonyl group in the main chain of the BACE1. And then I can also display a surface around this so that we can look at how the molecule fills the pockets. And so you see that the substituent really goes back into this pocket very nicely. Exit there. Now, another thing is that I was very interested in this pose here. This is simply a bromine, and it scores very well. It gets a really nice overlay with the ligand here, as you'll see when I bring this up. So it overlays very nicely, and so we've kind of reproduced that crystal structure pose. I also kind of looked at how these substituents are scoring. So I set up 3 queries to select molecules that are either ortho substituted, meta, or para around that phenyl ring. And then here I'm plotting the AutoDock score versus the counts of the blue bars versus the counts of the selected molecules here. And so you can see in the ortho substituents, which are shown here in red, they had a distribution that was much lower than kind of the average distribution or the distribution of the rest of the molecules here. So these are kind of low scoring. And you also see that in the table where the average is minus 6.5. If we look at the meta position, the average score is minus 8.0, and you can see that that distribution scores is much higher than kind of the average one here. Then similarly, the para was pretty high too, with an average of negative 7.9, but there were only 12 compounds in there. So you would, based on the best and the average, you can definitely say that the meta position is where you want these molecules substituted. That would be kind of the first round of AIDD. And then what I did was I took this as a seed molecule, and I set up the run so that it would optimize this ring out here. So that would allow for nitrogen or carbon aromatic atoms within the 6-membered ring. Would also allow certain substituents around the ring here. Then I held that fixed in the oxazine ring and optimized this phenyl ring, and then finally optimize the constituents around this oxazine ring while keeping the main oxazine ring the same. And in just those 3 runs, I was able to pretty much reproduce what the Rambots article selected as the best molecule and a candidate that they were going to probably push towards clinical trials in there. This last slide summarizes the talk. I started with a background of computer-aided drug design. The focus of the talk is the AIDD module in ADMET Predictor. It uses chemically intelligent Smirks transformations to generate candidate molecules, which can be scored in several different ways. One way is synthetic feasibility. You can use QSAR models. We can use what's called ADMET Risk. So these are the ADMET liabilities. We can also use pharmacokinetic properties, and we can use external programs to compute the objective functions. For the models, we penalize out-of-scope predictions, and we can use capping values on all of the models also. And then finally, I discussed how the program selects compounds for the next round of optimization using a Pareto optimal algorithm. And then finally, I demonstrated the program using BACE1 and docking into those as one of the objective functions, as well as ADMET liability, a BBB risk model, ADMET risk, and synthetic feasibility. I'm Michael Lawless, and I appreciate you attending my talk on AI-driven lead discovery.